Hey, my name's Lauren. I'm really interested in biotech and neuro-oncology, specifically seeing how we can improve patient outcomes with artificial intelligence. So today I'm going to be sharing a pretty recent paper or preprint of a paper coming out of the AIM program at Harvard, and it's so fascinating to me, and I wanted to share some insights and try to break it down a little bit so you can kind of get all excited about this technology and see the implications that it could have in improving patient outcomes. So as you can see, this is all about molecular subtyping, non-invasive, obviously, is here. And it's kind of showing how we can do this in different environments of machine learning. So there's going to be like self-supervised learning, and then there's also that is the transfer learning through the self-supervised learning approaches, but we'll dive into that more. So basically, the main kind of objective for this paper was to figure out a way to take the performance currently and look at, so I guess the status quo is basically this mutation status and classification isn't totally done. It's not really automated at all. And if it is automated, it has to be externally validated, and it lacks that in a lot of the clinical settings or just in the research beforehand. So not only were they were trying to increase the validity of it and also have it a multi-institutional kind of approach, but they also wanted to have one improve the workflow. So it wasn't just going to be from the scans, but it would be, as it says here, scan to prediction, so complete timeline, and having it all kind of automated within that machine learning. So obviously that comes with a lot of kind of challenges in what you would have to kind of integrate into the system. Not saying that it would be harder to manually, like it would still be more feasible to do this than if you were to do it manually. But this kind of comes really in handy because the intensive kind of strategy within all the different classification models and everything actually ends up being a really positive outcome in the end, because it can help for situations where you might not have enough data points to go from, or you might have very few of a specific nature. And that niche nature of the data types, as it'll talk on a little bit later, will show how for these tumors especially, even for low-grade gliomas, just in general, their nature from like a neuroanatomical structure, from neuroanatomy basically, they are very invasive. And resection, while sometimes that's not even optimal, it is basically kind of the main thing that has been a way to get a clear biomarker for, or clear pathology of, to see if these like tumors are actually of this BRAF mutational status. BRAF is a part of the cell signaling pathway, and in some cases, when it mutates, it's encoded with a protein. And instead of going in the traditional route through the cell signaling pathway, these mutated cells are growing and dividing uncontrollably, independent from the typical growth signal. And this can often lead to ineffectiveness within different therapeutics. Okay, so through this deep learning pipeline, there, is the two-stage process. We had our 3D tumor segmentation, and then the extraction of the axal tumor images, and then after that, it followed a slice-wise deep learning classification based on the mutational status, and the mutational status that they were looking at was the BRAF mutation. So there was either the point mutation, the fusion, or the wild type through this right here. And then these three are basically the ones that had this molecular classification and segregation in order to create the most optimal or accurate treatment selection and then risk minimization. So in this, the presentation of one of these mutations is actually clinically relevant with a lot of poor outcomes towards achieving this kind of end goal. So here we go into, deep learning, forefront of technology, medical imaging, and it has different applications within tumor segmentation, the outcome prediction, molecular classification. But the problem is this requires machine learning, even to just in general, requires a lot of data in order to train these models, right? So if we put this in an environment that we may have a lot of different co-founding factors that we might not be able to put into these machines. So the main issue was the lack of possibility for this to function in a limited data scenario. And this is like a main barrier towards applying deep learning for pediatric low-grade gliomas, because there's such lack of data just for the tumor cases in general. Because of this, it makes the mutation classification even more challenging, because the resources don't actually exist to even train the models, because the cases are so niche. So within that has a barrier to creating like more clinical efficiency. And then also to, if we're going to be using the algorithms that are like pre-existing, a lot of them require like the manual segmentation, which is, as it mentions here, you know, resource intensive, requires like specialized different team members. Also to just the facilities of like having this work within different geographical locations, right? So some hospitals or medical institutions might not have access to these resources or to these facilities that are maybe required to run these different complicated kind of machine learning algorithm techniques. So how can we basically not necessarily just create synthetic data, but create valid data without having the kind of pre-existing input? I like to kind of think about this where it's almost like you're lost in a maze, and you have your starting point and you have your end point. And imagine Google Maps didn't exist and you didn't have a map to get to figure out how to get to the end point. And instead of going from the beginning to the end, it's almost like you were just dropped in the middle of this location. And if you did technically have a geographical kind of like landscape of where to map and where to go, being dropped either in the middle, you would have no kind of idea on how to alter that, right? Because you're just stuck in a completely new, almost like area, right? You're not maybe sure the whole entire parameters. Again, this is just me metaphorically speaking. For those maybe who are much better with locations, that might not be the case. But in this perspective, just for this analogy, I feel like that's kind of the one approach that using like self-supervised learning through transfer learning, where it uses self-supervised and unsupervised learning, is like so relevant to this. So the difference between them is that transfer learning, even though it uses like supervised or unsupervised learning, these strategies technically tend to rely on a lot of like manually annotated labels to learn like the representation of such data. And then the self-supervised learning data, it's like using this supervision through the self-generated labels to create that, right? So it's self-learning. And then this is evidently like favorable in areas where you have limited access to incorporate the pre-trained data. And then to improve the efficiency of these cases, you can include a little bit of like genomic data in the beginning, or figure out a way to kind of integrate that before and afterwards to just test the whole validity of it. And they go into that saying too, and again here, which is just so crucial, why we don't want just a single institution to have this. And the few studies that are out, not this one in particular, but the one that were out before they launched this one, had the issue of that. So they're like, let's address this through using deep learning and having this complete workflow pipeline. So like here we have our pediatric low-grade glioma segmentation, our BRAF mutation classifiers, consensus block to predict the BRAF mutation status, and then we're going to leverage this through our developmental data set. After this, they put in the domain transfer learning using self-supervision approach. This is where transfer X comes in, and we're going to figure out a way to figure out if our tumor and the center of mass for the heat map is like relevant. So here are just some of the basic statistics that they had for the patient groups that they use. Beautiful diagram, which is basically our whole workflow overview. You can see here we have the beginning through just the MRI images and then the tumor segmentation. And then after it goes into the slice extraction. So then we have like our training block and then the consensus decision block and then the kind of resulting. So for the transfer X training block, which is within this, you can see that we have our pre-trained data, 2 sets, and then just cross-training and fine-tuning within the results of that. Here we have our 3 different outcomes within the classifiers in the subtype, and then so that is coming directly after we have our tumor segmentation, which is coming from another trained model. And then after that slice extraction is coming into here, we have our 3 different types, which then leads into our consensus decision-making block. And then here is just showing the model architecture of the different classifiers. So the mutation class outputs are where the input is either first checked for the wild type or the non-BRAF first. And then if it doesn't belong to any of those, or the non-BRAF, it will check like the logic to see if it has any BRAF mutation class with or without the other mutations. So while 3D tumor segmentation like already existed before this, what they really did is they were like, how can we give this pre-trained data and then also average with the pool of the ResNet 50 to replace it. They added a final layer of the single neurons for the binary classification, and then I think what was really cool too is their consensus was to have like the decision block as like a satisfaction to kind of get like it's almost like a litmus test if this is a best fit. And then within that, they were like, how can we make it more testable to exposure? So they combined through the different prediction models. They were like, let's cross train this to minimize the loss kind of on the axial slice development. So they tested this within the internal validation set, and it was 25% of the data randomly selected. And then in order to even add another level of accuracy, they put on this performance eval. So on each MRI slice, they were factored into kind of taking the average of the multiple like tumor slices that they ended up using. So to make sure like these scores were averaged out to give like a probable patient score, they did this with the threshold, and then here's just a calculation they did, and then after that, like the characteristic, they calculated like the area under the curve, which had the accuracy, and then that weighted output gave the different kind of output of the 3 mutational subtype classifiers. The results, the external test, so all patients, just it's clear to make sure that all patients did have a clinically diagnosed grade 1 or 2 low-grade glioma with like the different histological subtypes and intracranial locations. But for the developmental data set, they had 50. So the main kind of thing to take away here is that the tumor location not only can employ as the variable to perform this molecular subtype classification, but it's like not the only one. So typically, we would only have to rely on surgical resection because it was through having this tumor location. But through this, we realized that we can train this with the data, and it's not the only kind of test to see if this actually is indicative of like the molecular subtype classification. And that is so cool because not only can they like put on our own linear like regression models and like training this, we can also create it. Like the machine has this whole kind of almost identity and learning. And I'm getting so excited talking about it because just think about like the performance and the lack of resource intensity for different like hospitals or communities that are maybe less fortunate or have barriers to access to care. The BRAF mutational status prediction with like the composite classification from the Transfer X model that they trained ended up having a 77% accuracy on the internal validation compared to the 73% accuracy from training from scratch, which is kind of insane. And all the training approaches that they're using with the Transfer X module were most accurate at identifying these BRAF fusion mutations, and then that was followed by the wild type, and then our second one, which is our V600E. So, as some of the results from this study, we were able to see that not only did their scan to prediction pipeline workflow with deep learning had more accurate model attention, especially in addition to like if you were to do a manual tumor segmentation, it also yielded like the best averages between all of the classification tasks after it was going through the different regression models and the classifiers to see if the accuracy would be up to the standard of that in previous kind of metrics and studies before. So the prospective like future looking into how we could integrate that into maybe care, you could think about one, how would this help us train, future physicians, maybe like myself, and get familiarized with more contextually like different or niche cases, especially in practices and especially in the world of pediatrics and pediatric neuro-oncology. A lot of these cases may be selective to certain different like hospitals or facilities that can only treat these kind of niche conditions, or working on this clinical trial research. So having like an open source platform, and with a lot of the data that they used for this study in particularly, a lot of it's open source on GitHub, and training these models is a kind of main discussion point that we looked into earlier. The whole kind of reason for this was because a lot of gliomas, and especially pediatric gliomas, the locations don't allow for any resection. And if you're going to do a resection, it kind of poses a lot of negative risks on the patient, and it's actually not really worth the whole possible like complications if you go through it with the surgery. And then even through like the biopsies, it could be like comorbid and have like negative effects for the patient onwards because of this location of these tumors. So because of that, we have to kind of figure out what's a way that we can mitigate the effects for the patient, so they have the least amount of like negative effects and least amount of harm done, in regards to just even testing it to see how we could develop therapeutics for their tumor. Because we're not even in the whole kind of biopsy and resection stage, we're not even really looking to implement like direct drug delivery. It's just more to kind of get a gauge of what is like the tumor composition, and how can we use like the tissue sample to test for different genetic like predispositions and see how we can tailor our therapeutics. But if you're not even able to kind of get that resection through that, or if you get the resection, it might not even be that optimal to even apply to this. So that's kind of something to keep in the back of our head. And I really love what this study is, instead of kind of just having that as an unknown answer, we're using machine learning and having the unknown answers applied through the different classification systems and using like self-supervised learning, and having that taught through the transfer learning algorithms. So essentially, as a kind of conclusion point, like if we're able to have non-invasive detection protocols for BRAF mutation statuses with diagnostic imaging, not only could it help like targeted therapies that act especially on like the BRAF pathway that I was showing in the other clip, but it can also like help the enrollment in clinical trials. I'm not exactly sure the exact specific statistic for pediatric clinical trials, but I know for neuro-oncology and glioblastoma patients, which is more in adults, it's like 11%. And a lot of patient populations aren't joining them. And it's nothing on the patients, but I think a lot of it could be the lack of maybe education we have towards it, or the lack of having a positive like benefit from the clinical trials, because we don't maybe have enough targeted therapeutics. But through this and looking and analyzing these BRAF mutational statuses and seeing how we can actually integrate that with machine learning is going to be incredible in the next few years. It's incredible even now, but I get so excited talking about it. And then, so while it could also be used in things where tissue diagnosis doesn't work, you could also use it to compare it with like other malignancies, with like AI. It's saying here, so kind of overall showing that, transfer learning and self-supervision could really help develop a high-performing model and maintains good performance, not only on its actual data that it's training, but on external testing too, despite like the different tumor genetic types, and if it's a heterogeneous tumor or if it's a homogeneous tumor, you can kind of figure out more or less what the scanner and the tumor composition characteristics are, and then to kind of even have it more validation, to make it trustworthy and have like actual prosperity, hopefully in more clinical settings than are outside of just this institution, they applied their kind of metric evaluation model to get that anatomical like correlation to see if it's trustworthy. And then through that, that's kind of like another validation point. So here the accuracy is similar, like very close between the internal and external, which is good to say the least. And essentially kind of having it, not only will it help enable like specific patients for these different BRAF inhibitors to hopefully have better or progression-free survival than chemotherapy, the differences within, the performance degradation could also be more between the lack of like MRI parameters and like different scenarios in data with the institutions, and maybe less of like an actual lack of the data set, like the data sets working. But again, for this, because it's like more practical, since it doesn't rely on any of this manual segmentation, it is just like so much more feasible for these kind of clinical settings, and just so many other opportunities for treatment options. And what I found really interesting here is not only does it perform better and more accuracy, similar accuracy with like the internal and the external data sets, it also is able to like highly perform and be reliable for classifying tumors in like neuroanatomical regions that are more challenging if we were to do an intraoperative like biopsy. So these are like the optic pathway, the thalamus, the brainstem, which is insane because not only does this help physicians and treatment teams make like more informed decisions, it can also again help with more targeted therapies, even in spite of not having a tissue diagnostic evidence or anything like that, if it's just completely unattainable. So here we kind of have the different representations through the pipelines, and this is kind of how the consensus decision block worked. And then you can see the different tumor lesions in these T2-weighted images, and they're all highlighted with the arrows. So here is one of the tumor lesion areas, and then you could see here, again, here, and here. And then same with these ones. And this is what it applied through the prediction of the scan to prediction pipeline on these external data sets. So again, it has their 3 subtype classifiers, your wild type, your BRAF fusion, and then your BRAF V600E. And then, pulled together, it'll make the decision block and baited with there. So you have like all these kind of results. And aside from this, in the other studies that had mutational classification, again, most of them were developed with this manual segmentation, like backgrounds, or it had like pre-engineered radiomics, which basically means that like if this was going to apply to an external data set, it would just fail. It would completely like be, it just wouldn't work. It wouldn't be any tips to the patient populations or any data because it's trained specifically on that data. But if you make an algorithm system that's not trained exactly through those first like 1,000 data sets, which is very limited, then you can apply like external validation into it and actually giving it more unique patient populations or more just like not familiarized like content, and it'll teach itself, it'll adapt, and it'll pivot based on the kind of familiarity that it has. So I think another thing too that I found that was really cool is, so you can, use neural networks to outperform the like prior classification outcomes, especially with like the area under the curve, because it was affected by like the size of the data used in the training. But like doing end-to-end deep learning, as I said, is feasible even in a low data setting, which is insane because you can use the interclass cross training combined with the transfer learning. So you don't even have to manufacture even more patient kind of data or any, well, you wouldn't manufacture to begin with, but just it kind of completely removes that barrier to access or to receiving care within those areas. And then so actually having like the assumptions of the different like categories within it can use that task to like add it into the feature representations. And I think even with this, like applying the consensus decision logic, they said to like the multiple binary classifiers can also help to mitigate overfitting for the limited data set. So this is talking about the interpretability and how it's very important for like clinical translation of deep learning, and how through the GradChem approach that they used they tried to conquer the only thing of having case-by-case visualizations for like the end user, because you weren't able to get a model and overall. So they were using their evaluation metric. So they used like the spatial knowledge through the tumor, auto tumor segmentation, and then they use it to quantify. They found the distance, and then the model detention, like basically where it was in the region of the interest in the tumor, they could provide it with the clinical like knowledge and to see if it's valid or not. So not only are you kind of having the integration of the physician or the person of care, you can also kind of integrate it with the intratumoral like information, which is so cool too. But again, yeah, this metric can be reported case by case, or if it needs to kind of overrule a data set to like compare attention to the different models, it can also do that. And again, this will only kind of improve more accuracy as this technology develops and further progresses. So here is kind of some limitations and maybe how we could combat them. So the main kind of thing was saying when it's like subjective with the biases of the patient samples. And then this was kind of a solution to combat this was through having like the blinded external validation set, but to kind of even encourage more independent validation for these results, they're hoping to kind of have a pipeline that would be based on other images that could enhance the performance. So for this one, because it was more, again, pediatric low-grade gliomas, they use primarily like T2 weighted MRI imaging scans. So if they were to add like the FLAIR or the T1 and the diffusion weighted MRI, or even like one with like a contrast enhanced, that could even help improve the accuracy. But because so far the only kind of available diagnostic sequence for pediatric low-grade gliomas with like the contrast enhanced was the T2 weighted images. So this again could be another option to kind of look into further and seeing how this could apply maybe to other gliomas of like non-pediatric patient populations to see if those diagnostic like sequences are more readily available. And then they use the 2D approach with the slice averaging to like minimize the overfitting in their limited data set, but with like further collection, if they were to do so, you could possibly see like the implications of a 3D approach and how that could maybe work more effectively. But again, it would like increase the model parameter size and then make it like more prone to overfitting. So that could be a downside. And yeah, essentially, so they developed externally validated an imaging-based scan to prediction pipeline to analyze T2 weighted MRI as input and output as the BRAF mutational subtype for pediatric low-grade glioma. They used a novel of transfer learning, self-supervision, and to mitigate the overfitting. And they were able to assess like the performance and interpretability of AI imaging. And basically, it warrants the whole like prospective validation to determine if this could be used in clinical settings where like the tissue and genomic testing is unavailable, or if they're both unavailable, or one or the other. And that's kind of my last thing that I wrote here is like next steps. How can we find possible use cases for this technology and maybe think about how would we train different hospital systems to integrate this into their care, or if you were going to do so, like what would that look like in maybe from an education standpoint, and then also to like where is more of the data generation coming from, and then how can we like train younger physicians to understand this, but then also use more genomic kind of biomarkers to back up it like they were using here, and also kind of understand how we could maybe build a 3D type with the 3D approach instead of just doing a like 2D approach for the slice like averaging. Hey, thank you so much for reaching the end of this video. It means a lot if you watch it to the very end. I hope you learned something. I just want to say I'm still a student. I'm an undergrad, first year. So if there were areas that I maybe didn't interpret correctly or didn't understand, feel free. I'm totally open to any feedback, and I'm so excited about exploring this space. So if you're one of the authors of this paper, I would love to get in contact, maybe share this with your lab, and I hope I was able to kind of do your work justice because it is so exciting to me, and this makes me beyond thrilled and excited for AI and medicine, especially like neuro-oncology. And you can see on my face, I'm like so excited, and I cannot wait to see what the future of this research field kind of has to offer.